Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working through Chapter 13, which is viruses. So we have described a viral structure, and then we've described bacteriophage, defined bacteriophage, and went through the lytic cycle and the lysogenic cycle. So up until now, we've been talking about bacteriophage, viruses that infect bacteria. But this is medical microbiology. We want to know about viruses that infect us, right, and cause diseases. So we're going to move on from viruses, bacteriophage, viruses that infect bacteria, and move on to viruses that infect animal cells or eukaryotic cells. So I do need you in your mind to compartmentalize those because I have students that mix these up. The stuff we learn about bacteriophage is useful because it's a good model. Again, learning something in a simple form, then when we start learning in the more complicated form, we've got that simple form to base it off of. So it's a good model for learning about animal viruses, but a eukaryotic cell is a lot more complicated, which means a virus that infects a eukaryotic cell has a lot more things going on. So put a line into bacteriophage, which we've done before, and then moving on now into animal viruses, viruses that infect eukaryotic cells. So let's look at that, how they're similar, but then look at the differences from that. Okay, so we said it's a good model, so that's good. And the good news is it's the same steps have to happen. You still need attachment. You still need entry. You still need synthesis and assembly, and you still need release. So again, it's a good model, but we're going to see how each of those things vary a little bit in the animal viruses from that. All right, so let's look at attachment first. We've got to get attachment. All right, good news, a lot of similarities. It is still protein-protein interactions. It is still glycoproteins or spikes, and those are just different words for the same thing. It depends on the literature goes back and forth. So glycoproteins or spikes is a protein on the virus that is going to bind to a protein on the cell. So they'll talk about it as a receptor. And remember, it's just a protein that a cell is using that virus has been able to attach to and use it as a way to get in, okay? So exact fit protein-protein interactions between glycoproteins or spikes and proteins on the cell surface or receptors is needed. So that's how it gets in. And again, we said enveloped viruses or capsids, nucleocapsids, non-enveloped or naked viruses, whatever's on the outside is where the attachment proteins are located, and they can get in as well. All right, so if a cell does not have the right protein receptor, then that virus can't get in. So eukaryotic cells, a lot of variety between eukaryotic cells, which means some viruses can only get in certain types of eukaryotic cells. This is called the host range. When the protein-protein interactions, they drive which cells can become a host. And so this is known as the host range. Now, some of these glycoproteins are going to use a cell receptor that may be very specific to a certain type of cell. An example, hepatitis B, that's a viral disease. It's caused by a virus, and that virus only infects human liver cells. The receptor needed is only found on liver cells, and in fact, only in human liver cells, not primate liver cells or any other type of mammal. This is a very specific host range. So we consider this a very narrow host range. Now, there are other viruses that have a more moderate host range or even a broader host range. Polio viruses, they infect a couple of different types of cells in our body. They can infect intestinal cells and we can infect nerve cells. And whichever cells get infected, that's where we see the signs and symptoms. If I go back to hepatitis, hepatitis only infects human liver cells, and you notice that the the signs and symptoms of hepatitis include damage to the liver, right? Jaundice leading to jaundice. And this is because only the liver cells are being damaged. So in polio, they can infect intestinal cells and nerve cells, and so you get different types of damage from that. And not only can polio affect human nerve cells and intestinal cells, it infects primates. So some of our close relatives, the primates, can also come down with polio. Rabies can infect all mammals, affect all mammals. So the rabies virus um, attaches to a receptor protein that is found on all mammalian cells. I think there's an exception. I think armadillos can't get rabies. I don't know. There's some exception. You guys can fact check me. But all mammals can get it, which is your dogs, your bats, your raccoons, all of those types of things can get rabies. And that's a more broad host range. Okay, so attachment and then understanding what host range is about. So after attachment, what comes next? We need to get inside. That's the same is true for animal viruses. Now, if you remember bacteriophage, they do that injection method, right? The ghost capsid is left on the outside. The nucleic acid is the only thing that enters. Eukaryotic cells are more complicated, and there's a couple of different ways that entry can occur. So different viruses, different types of viruses use one of these three different methods to get in. So if it uses this method, if the instructions inside the virion says it will use direct penetration, that virus will always use direct penetration. So it's not a pick and choose type of thing. It depends on whatever it has the instructions, the nucleic acid, the DNA that gives it the instructions on how to get in. So we have seen three different ways that different virions can get inside animal viruses. So let's go through these. Direct penetration, membrane fusion versus endocytosis. So let's do direct penetration first. Direct penetration is when you have a nucleocapsid of virion. It binds to a receptor on the cytoplasmic membrane, and when that happens, it causes the entire capsid, nucleocapsid, to kind of sink into this phospholipid bilayer, kind of sinks into that membrane, kind of creates almost like a hole in that. Once it gets embedded or penetrated into that membrane, the capsid will actually break apart. And that capsid breaking apart releases the nucleic acid, the virus, inside the cell. Now, vocabulary term, when a capsid opens up, that's called uncoating. So we don't see that injection that we saw with bacteriophage. We see an uncoating, and then just the nucleic acid enters the cell. Now, this direct penetration only occurs with a non-enveloped or a naked virus. Enveloped viruses cannot use this method of getting in. So that only happens with a non-enveloped virus. This is direct penetration. All right, let's do the next one, direct penetration. What else? There's another entry method called membrane fusion. So if we're talking about fusion, lots of different fusions, right? Different food fusions that are really popular right now. If we're talking about fusing something, we need at least two things, right? So if we're talking about membrane fusion, we need two membranes. One of those is going to be the host membrane. So that's one of the membranes. The second membrane is what? That enveloped virus. So membrane fusion is a type of entry that only can happen to enveloped viruses. A nucleocapsid without a membrane, a naked virus, or a non-enveloped virus cannot use this method of entry. So membrane fusion, what happens? So we still have protein-protein interactions. We still get that attachment, but this attachment causes these two membranes, the envelope and the host cell membrane, to fuse. Phospholipid bilayers, remember they can move around, that fluid mosaic model. They move around, they attach, and they actually attach, and these membranes fuse. And you can see that viral envelope is left on the outside of the cell. So what enters the cell? The entire nucleocapsid enters the cell. The capsid plus the nucleic acid enters the cell. Now this is the first method we've seen where the capsid enters as well. So the nucleocapsid enters the cell. We still need to get the nucleic acid out. So uncoating, that breaking apart of the capsid, still occurs. It occurs this time in the cytoplasm, and then the viral genome, the nucleic acid is released into the cytoplasm. Okay, so direct penetration, only non-enveloped viruses. Membrane fusion, only enveloped viruses. The third method of entry that we observe in animal viruses is a type of endocytosis. Okay, you know that word, right? Endocytosis, bringing in something inside the cell. Cells use this to bring in nutrients or other things that it may need that are too large to go through. Endocytosis happens when, again, you get attachment, that protein-protein interaction, and it causes the cell to undergo endocytosis. So the cell is kind of tricked into bringing the entire virion into the cell. Now, this can happen whether it's a enveloped virus or a non-enveloped virus. So in this particular picture, we see an enveloped virus is undergoing endocytosis, but non-enveloped viruses can also use this method. Some non-enveloped viruses also use this method to get in. All right, so what comes in? Now, not only do you have the nucleocapsid that comes in, but it comes in with or without the membrane, depending on whether it started with a membrane or not, but it's also within a vesicle. Remember eukaryotic cells and that vesicle system where it's covered by that? So we got a lot of releasing that needs to happen. So the vesicle will break open, the capsid will uncoat, and eventually the nucleic acid gets released into the cytoplasm. Okay? All right, so those are 3 methods of entry. I'm going to stop the video here. Make sure you're talking about animal viruses. So don't get confused with the lytic and lysogenic cycle. These are methods of entry used by animal viruses to enter a eukaryotic cell. All right, see you in the next video.